following is a conversation with Dr. Johan van Hylckama Vlieg, the Chief Scientific Officer at Freya Biosciences, one of the world's leading microbiome companies focused on women's health. Johan is a pioneer and a successful entrepreneur, as well as being an esteemed scientist within the field of gut microbiome research. He's had a successful career in industry, being involved in the creation of companies, raised significant amounts of venture capital on management teams, and has published really impactful research in the field. I've been wanting to get Johan on the podcast pretty much since it started, and I was really excited about having him on. The work that he's doing alongside Kaleido and the team at Freya Biosciences, I think, has massive potential. You'll hear about the concept of vaginal microbiota transplantation, which is very different to the fecal microbiota transplantation we talk about normally. In many respects, though, it's quite similar insofar as the procedures associated with finding a donor are similar, and the concept of re-establishing the microbiome through healthy microorganisms in someone suffering from a disease or infection is the same. You can expect to learn about entrepreneurial journeys in this podcast episode and what it's like to be a biotech executive making difficult decisions. Johan also touched briefly on some of the work that he's doing with the Gates Foundation as well, who are big proponents of the microbiome's importance in maternal health and also the developing world. Thanks again for your support for the podcast. We've had some amazing guests on recently, and we plan to continue to do that over the course of this year. Why don't we start with, why are you excited about the potential of the microbiome?
So first, James, I'm really happy to be here. And I think it's the fact that you say that you've been following me for all of your life also is a testimony on how old I'm getting, probably. But let's not go there. No, why am I excited about the field? I was excited about the field 20 years ago. When we saw these first experiments coming out of the lab of Jeff Gordon, these famous experiments that you could essentially transfer the obese phenotype in mice through the microbiota, right? That was the first time that I thought, oh my God, this is real. This has therapeutic potential.
Yeah.
And since then, we've come an enormous way, right? Because we know so much now about not only where the microbiota has an impact on host health, on human health, but also very much by what mechanisms, right? It's much less of a black box as it was then. It's often very precise. We know which microbes, what metabolites they produce, or what proteins they produce, how they interact with specific host receptors, how they impact inflammation, metabolism. And I think that is just breathtaking. And I think few of us early in the field could have even dreamt that it would be so impactful and have such wide-reaching potential.
Yeah, and when we were doing the prep for this podcast, one of the themes that's really stuck with me was you mentioned that the science has absolutely delivered, but the translation has maybe not delivered as well as we would have liked. Could you just unpack that concept for the listeners a little bit and explain, how have we progressed from a scientific perspective, but maybe why the translational aspect hasn't worked so well? And then that should segue into the exciting work that you're doing in the clinic.
Yeah. So I think if you would take the perspective of investors and drug developers at Big Pharma, and you mentioned, well, I work in the microbiome, doing drug development or working on interventions, they will have a sort of, okay, yeah, right. This was very interesting a while ago, but it hasn't actually delivered yet, right? Which, by the way, is not unusual for new modalities. I think we often see that. All these mRNA therapies also have, I think, gone through this phase.
It's true.
And I think there are a number of reasons for this. And I think the first thing is that it is a very complex area, right? It's not that we're talking about a specific drug target, a receptor that we want to modulate through that have an effect. We're actually talking about the interaction between two living entities. And then the microbial entity is not just one often, it's actually a whole ecosystem. So the mechanistic complexity is really, really high. And I think some of the early interventions that people try to develop were maybe a little bit, maybe the underlying scientific assumptions were a bit bullish, right? So People wanted to move very fast. Big VCs didn't want to miss the train that was leaving the station. And I think many of these earlier days were maybe, to some extent, a bit naive even. And then, if you progress and you don't show success, then you fail, right? And so there are really quite some people in the field that I think got hurt, who invested early on in the early 2010, 12, 14. And then, companies failed, right? So I think that's
just one point there. Sorry to interrupt. Back in 2010, 2014, could you just explain to the listener, was the microbiome the in thing and everyone wanted to be a part of it? And what was the trigger for that?
I think for a while, let's say until deep in the 90s, I think the microbiome, or the microbiota as people called it, was actually not a very popular thing. There were a few scientists that were doing pioneering work, but typically, as I often say, it was one parallel session 2B in a big microbiology conference, right? Yeah. And I think that really changed by a few events. One was what I just alluded to, that work of Jeff Gordon, who is definitely a pioneer in the field. And the other big inflection point there was, and this you know very, very well because you're active in this area, is the use of fecal microbial transplants. The fact that these early studies showed that you actually could cure, not just mitigate, cure a deadly infectious disease. And then these trials that even had to be canceled because they were so successful that it would be unethical to not give everybody the drug, right? So I think
rare.
that is really, really rare. And that generated this explosion of interest. And all of a sudden, I think it was 2012, for instance, the microbiota was on the cover of The Economist, right? So man, who would have thought that this would ever happen? So I was on holidays actually, and I posted on holidays. I couldn't believe that this had happened. So, and then many of the first, let's say, company activities, they focused on transplants, fecal microbial transplants. And I think one of the other things that maybe in hindsight went a little bit was unfortunate is that almost everybody focused exactly on this specific disease, recurrent refractive Clostridium difficile infection. And everybody pursued this. And all the VCs that supported these companies wanted them also to focus on this specific indication. So that meant that a lot of the money that went into the field actually went on this specific infectious disease. And then the market in the end is small and there's only one or two winners.
Exactly.
And I think that hampered the field as well. While maybe and this is also my conviction, the biggest opportunity is probably in immune-driven disease, right? And let me try to maybe briefly explain what I mean with this.
Yeah, please.
I think it is extensively documented now that our microbial residents in the gut or elsewhere, they have a massive impact on what our immune tone is, right? Whether we have inflammatory pathways, the whole immune system is very much geared to keeping invaders, bacteria, microbes out. And of course, because of that, it's very much responding to microbial signals. And I think we're actually at the point now that it's fair to say that it's almost unthinkable to work on a immunotherapy without taking to some extent the microbiome or microbiota into account, right? Because it sets so much of the context. And as we also discussed in the preparation, for me the maybe most breathtaking area in this field is actually what we started to see as of 2018. The fact that this very potent and groundbreaking class of therapies in oncology, ICI, immune checkpoint inhibitors, where many people respond, but also in most cancers, actually most people don't respond. Yeah. The fact whether you will respond to this or not is actually very much governed by your microbiome. And by intervening on the microbiome, you can change the immune checkpoint inhibitor response. And this is all immune related signaling. And that holds for every area almost. And it holds for IBD. If we talk about the gut, it holds for skin. And it also holds for the area that I'm specifically focusing right now is women's health.
And women's health.
Diseases of the female reproductive tract.
Before we get on to Freya and all that exciting work, can we just go back a step? The sort of huge flurry of activity that we saw around about 2010 in C. diff. So, you're someone who's co-founded companies, you're building companies as an executive. What do you think was the driver for all of these companies pursuing C. difficile infection when indeed relatively small patient population, there was already existing standard of care for the kind of acute phase of the treatment? What was the pitch that got all of these investors excited, do you think, given that it wasn't like, an enormous patient population?
Yeah. So just to be clear, I have never pitched a C. diff product, I think. So, but I can sort of try to build the argumentation that I think people used at that time. So first of all, because of that specific study in the New England Journal of Medicine that had to be aborted because it was so successful, it generally, I think, was considered like a low bar, easy. This would work for sure, right? And if you have a new modality, you would like to go into an area where it's not too complicated and where, because of the not too big target population, you probably have an accelerated regulatory path as well.
Fair.
So it felt like a very obvious thing to do, I think, for people then. And then almost every company also had on their pipeline multiple other assets in these really, really large indications. Right?
Yes.
But then as they became cash constrained and it proved a bit more difficult than everybody anticipated, what happened is that the boards and the VCs and the funders, they said, well, now you have to really, really focus on the core. And it's your C. diff product. Make it happen. And the leading companies were very much focusing on that at the time.
That makes a lot of sense. But one of the crazy things for me, though, is if we go back to that period of time, this RCT in the New England Journal of Medicine catalyzed pharma deals, blue chip investors, company creation, the whole academic grants, the whole lot. And that's great. But then you just described transformational impact in immunotherapy. And these immunotherapy drugs are arguably, I think they are the best-selling drugs in the world. Keytruda as the number one example, 10 plus billion a year of annual sales. Why do you think that there's not investors jumping and pharma companies jumping at this next frontier, so to speak, where there's been some proof of concept from FMT in immunotherapy, but there hasn't been the same kind of explosion of interest as we saw back in 2010, 2014?
Yeah. That's a very good question. So first, I will start by saying that I actually share your view. I also don't fully understand why there at least is not more enthusiasm on this, right? So I think it's justified. There are some activities. There are a couple of companies, I think some of them you maybe have had even on your podcast, that are active in this area. And especially once we get solid phase 2 data here, I think that will probably be another inflection point for the whole field.
Makes sense.
But I think there are also some dynamics there. I think we are in a stage right now that actually being a microbiome company or a microbiome therapy really is not a favor, right? And that is because people have been sort of hurt by this, and there's some intrinsic reservation to this. And I will also say this whole area of improving efficacy of immune checkpoint inhibitors, we think about it from the microbiome perspective. There's also other activities there that maybe you compete with. So, Fair enough. But let me be very clear. I remain very deeply convinced that at some moment, if we get convincing data in an indication other than an infectious disease, that can also very quickly reverse everything, right?
That's good. Because it feels like a bit of a breakthrough insofar as these studies that we're talking about, just to give the listeners some more context. Immunotherapy has been a complete game changer, hasn't it? It's treated cancers that are aggressive in a manner that we could have only dreamed of 30 years ago. But they don't work for everybody. In fact, quite a significant percentage of patients don't respond. And what these studies showed was could we call it kind of causative level evidence rather than correlative evidence, insofar as they actually took stool samples from responders, i.e. people who had a good response to immunotherapy, and moved them into the intestinal tract of the non-responders, which is not a classical FMT because you'd usually have a healthy donor. And some of these non-responders became complete responders. So it kind of feels like the perfect story. So we've had this kind of breakthrough outcome in the microbiome space, but I've not seen hundreds of immunotherapy companies popping up. I know some companies are doing it. So, I guess what I'm trying to say is it's not had the same impact as the early stages. And your thesis is we still have loads of reasons to be optimistic, but maybe we just need the industry to do a bigger study, more patients. Do you think that's kind of what's required to get things going again? Yeah.
Yeah. I think the really good message is also that I think the companies that are still active in the field, they feel, and I'm not talking just about Freya, I think in general, including your company, they feel much more mature. I think it's much more rooted in good clinical evidence, in solid thinking about what a clinical development path would be, in understanding actually what pharma needs to see from a clinical study, right? Because some of these early studies were also very science-driven, which is not bad in itself. But if you want to do drug development, there's other things that also need to be taken into account. So the whole field has been maturing, and maybe in 10 years from now, we will look back at this period with very little investment as actually a very healthy thing, because it sort of brought the ones that were doing good work forward, and the whole fact that it was so diluted over so many companies, all in their preclinical or preparing for a phase 1. Yeah, there's also a certain amount of money that a field can absorb to progress.
So is it fair to say then that, in your opinion, infectious disease was kind of the easy win, and now we're moving into harder, more complicated indications, but maybe more upside? Is that a fair summary of your thinking?
I'm not sure whether it's in general for infectious disease, but the ones especially where we focused on. And I also don't think it's an easy win, because we've also seen that these clinical trials are actually not as easy maybe as people initially thought, right? With respect to inclusion criteria, monitoring of the infection, product quality. We don't have to discuss that here in detail, but I think it proved, like for almost any drug, that conceptually it may look very simple, but once you want to make it into a product, there's always stuff that is more complicated, and you learn as you go, and we all learn from failures as well.
Indeed. So maybe this.
But we have two approved products. Let's not forget this. I think that is big. Yeah.
That is big. Maybe explain to the listener why it's so important. It sort of paves the way for others. It gives investors some certainty about this being a drug. Why is it really useful to have approved drugs for the kind of lay listener? Why is that a seminal moment for the field?
Yeah. So I think if you're a listener to this podcast, you're somewhat a believer, and you truly have interest in the microbiome, and you're convinced that this is going to be very, very relevant, right? But if you are out there in the bigger world of drug development, this is still a really odd modality, right? Because typically microbes you don't want to have in your product, right? It's contamination. So it always already starts there, and you're laughing. But if you talk to people in manufacturing and so forth, that's really what the first sort of gut reaction. So I think the fact that we have products approved sort of validates that this actually can be a product. And that means it can be clinically developed. There is a regulatory path. We should maybe also talk at some moment a little bit about that.
Sure.
And there's actually a business case to be made for a product like this. And today it's donor-derived. I think another inflection point will be when it's other products that are maybe defined consortia or proteins or metabolites from the microbiome. So, there's a lot of room for growth still for the field as well.
The sort of aseptic mindset that you're describing it really hits home for me. because, our product in particular, I think, given that it's derived from stool samples, is about as non-classical, typical as you can possibly imagine. And then when you're talking to people about facility design or CDMOs, you have to explain that actually, you're kind of protecting the product from the environment, even though that is full of microorganisms, because you only want those microorganisms. And to someone who's only worked on a small molecule, an injectable all their career, to even imagine that what you're thinking about as a CSO is how to make sure that there's as many microbes in there as possible. You're right, it's just totally different. So there's a lot of education to do. But this brings me on, I think, quite nicely to one of the key themes for today's discussion, which is women's health and the microbiome, and how it's kind of maybe a case study, if you like, and neglect, maybe not the right word, but it's being kind of underserved, I think, quite significantly. And you spoke very passionately about this when we had our kind of pre-podcast touch base. So I'd love to just have a really kind of open discussion about women's health and how it links to the microbiome, and that will then segue into the really innovative and exciting research that you're doing at Freya.
Yeah, no, I'd be happy to do that. So maybe let's start a little bit anecdotally. When we started with Freya and we wanted to raise money in 2017-18, the reaction that we got from VCs was, that is interesting, but we don't do women's health, or there was no strategy almost.
Wow.
Now, if we take the flip side, and we are here in 2025, that definitely has changed, right?
Yeah.
And I think there's maybe a couple of key points to highlight, which, if people are interested, I think is very well summarized in a report published by the McKinsey group, the consulting group, like a year ago, I think it was. It's called Gender Gaps in Women's Health, or I should have known the title precisely, but you'll find it if it's McKinsey.
We can put it in the post show notes.
Yeah,
we can get that. Yeah.
So, and it highlights like 4 main things that they analyzed when they looked at the market. Because these, of course, they typically will analyze whether there's a market opportunity there. And the first thing they notice is they say, this area the science is so immature. The study of the human biology defaults to the male body, actually. And, there's things like in clinical trials up to 30 years ago, people try to avoid having women because they had menstrual cycles and it gave noise, right? So, which is actually biologically relevant. So there's a lot of biology on specific female women's metabolism that is actually lacking. And that's not only for the female reproductive tract, but actually global metabolism. So there's really something to do there. Then the second thing they notice is that there's just very, very little data, right? Women's health burdens are systematically underreported. And again, that holds to specific things related to the female reproductive tract, but also to things like cardiometabolic disease and so forth. Then care delivery is also not fully developed. So women are much more likely to face barriers to care and experience diagnostic delays and so forth. And last but not least, there's no real investment. We have actually seen a period of divestment, for instance, from major pharma companies in women's health. And that is in itself, I think, quite mind-boggling, right? And I think, you could say almost scandalous. Now, there has been a real change in this, as I said. I think in 2025, it's a very different world. There's a lot of awareness. I think there are a lot of good initiatives. Some of these areas are much better addressed, especially that area of care delivery. There are some really, really good initiatives, platforms to ensure better diagnosis and better connection of patients to their care providers. And some of these are not only having a lot of patient impact, but actually are also commercial successes. But what for me as a let's say, intervention and drug developer is always amazes me is that this area of actual new product development is still very, very limited. There's a lot of talk, but not so much walk the talk there. So that's also why I think, why we at Freya are so, dedicated to actually drive activities in this area.
And Johan, for the listener, is that just, are we talking about women's health and microbiome, or are we talking about women's health full stop, across the board?
Women's health full stop, yeah.
Wow.
Yeah. So and it's maybe also good to highlight that while I think there has been consistently interest and activity in specific fertility-related activities, other activities that are much more, let's say, disabling or impacting quality of life, like endometriosis or postmenopausal symptoms or uterine fibroids, all these things that people hardly talk about, but that really, really impact the life of women. There's been hardly any activity for a long period. That's just starting now, I think.
Wow. So bring us back then to the start of Freya.
Yeah, so I think having sketched this context, what I think is really, really interesting is that many of these diseases that we could think of, fertility area, but also these more inflammation-driven diseases, they have this immunity component, right? And of course, the female reproductive tract also has its microbiome, the vaginal microbiome. So already during my time at Christian Hansen, which was one of the employers I used to work for, probiotic development, we actually, my scientific team, but the whole organization had a real interest in developing products in this area. But for a essentially vaginal microbiome targeting products, live biotherapeutic products to target the vaginal microbiome. And that, I will not deliberate too much on it, but that proved very difficult because it was the supplement area, and locally administered is difficult if you want to stay out of the drug regime. So we had already had an interest to do it in that context, but it was not the sort of business context to do it. So myself and a few other people, we actually said, well, let's then build a company in this area. So it really started like this as a evening and side project.
That's cool. Sorry, what was known about the vaginal microbiome then? Like, had we characterized it? How did it differ between the gut and the other microbiomes of the body? That could be quite interesting to understand.
I think it's a very important point you're bringing up. I think we should spend some time on this because that also, I think, explains part of the attractiveness for development of intervention products. So the gut microbiome, I think, as everybody on this podcast will know, is very complex. It has many, many organisms. There's a large subject-to-subject variation. It responds to diet. There's lots of things known about this, right? So the vaginal microbiome is unlike any other microbiome on the human body. And it's actually very unique even for humans, the way the vaginal microbiome is built up. And at the highest level, if you really want to do a reductionist approach, it basically exists in two sort of states, right? One is a state where you have lactobacillus dominance, so bacteria very related to the ones that we have in our fermented food products, but a different subset of those that are low complexity communities. There's a limited number of species. Well, you have different strains, but a limited number of species. It's generally related to mucus and barrier integrity. You have a low pH, and all these things help to get invading microbes that might be more detrimental out, right? The other sort of opposite state is what we will call, for simplicity, dysbiosis. And in many cases, this is characterized by a much higher diversity, right? And often facultative or even strictly anaerobic bacteria. You get biofilm formation. And interestingly, this dysbiotic state is across the board, has already for a while now been associated with worse health outcomes. That relates to symptoms of bacterial vaginosis, which is inflammation and discomfort and can even go worse. You have candidiasis as well. It gives a higher risk for infections, STIs. But it's also correlated to immune deficiencies and even in fertility issues, ranging from preterm birth to actually more difficulty to get pregnant. Now,
so just to understand, did I hear you right in saying the lower diversity vaginal microbiome is associated with a more healthy, broader phenotype, but actually a higher diversity vaginal microbiomes associated with more in the way of deleterious disease state. Is that what you said?
That is exactly what I was saying, yeah.
Because the keen listeners of the podcast will, of course, be very familiar with the concept of high diversity most often being a positive thing. So it's actually different. It
is, yeah, it's truly
very
different
interesting.
Yeah.
Very interesting. Okay. So that's a key core concept then for the listeners to take away there is a microbiome. It's usually more simple, and it's usually the bifido, I think you mentioned, and
it's mostly lactic acid bacteria. There's a few species, mainly Crispatus is often mentioned, but also Translike Gasseri. And then you have L. inus, which is a more like a less clear role, but it's lactobacilli.
Very, very interesting. Okay, sorry, I cut you off, but you were building that scientific basis for all the work that you're doing at Freya. So you're talking about what it is and the disease state. Yeah, thanks.
So the idea we had is let us identify the best vaginal microbiome intervention, but it's not about curing dysbiosis. In the end, you want to bring a benefit to the host, right? And I have already mentioned a couple of times the relevance of immune activation, immune pathways. So we set out to do two things. First, map the healthy women microbiome, vaginal microbiome, and do that by taking samples, do deep metagenomic sequencing so that we get really like strain level, genome level resolution. But in every sample that we took, we also did a host immune profiling. So in a cervical vaginal secretion, you can actually look with different technologies at the protein or at the transcriptome level at the activity of host immune pathways. Right? And we've done this now with over 1,000 women. And lo and behold, there is a very, very, very strong correlation between microbiome composition and the activation of specific immune pathways. And the overall picture that emerges from this is, if you have this lactobacilli, you have lower activity of inflammatory pathways, right? If you have dysbiosis, you get activation of many inflammatory pathways. There is some let's say, some granularity below this, which makes it, even possible to target specific pathways, but that's the high level picture. And to give you an idea, and this we also presented already at conferences, and it's actually also available at in a preprint manuscript that's on a preprint server at this moment. We've identified, for instance, that the complement pathways, there are 3 main complement pathways. This is one of these first-line defense pathways against infections, but it's also involved in many inflammation-related diseases. We've seen that if you are lactobacillus dominant, you have consistently a lower expression of most of these complement pathway genes. With a few exceptions, the genes that are higher expressed are actually the repressors, right? Which you would expect. So the genes that are involved in shutting things down. So that's sort of the cross-sectional element of this. And I think it's a really, really valuable resource. And I talk about this now very simply, but to do actually these measurements took us way more time than we anticipated, because many of these sampling methods and these analytical methods, they are made to analyze serum, right? And serum is very homogeneous in composition. It's always
not
the same
so noisy.
Also, always the same osmolarity value and so forth. With the CVS, cervical vaginal secretion, that's very different. So it was quite some work to do this. So then the second thing we said we would do is, okay, in order to really show causality, right? It's nice to show that inflammation is correlated to microbiome, but you actually want to do an intervention. And we decided to do to be inspired by these fecal microbial transplants and do vaginal microbial transplant studies. And we did that essentially in healthy women. So they did not report any vaginal symptoms. They were generally healthy. But we screened women for whether they were dysbiotic or not, right?
Okay.
And you see typically, depending on how you exactly define dysbiosis and what population, you see typically somewhere between 25, maybe a third, maybe a little bit higher of women actually is in this dysbiosis state, this high diversity state.
Just one question, Johan. Is it one of these diversity indices that you apply, which people might be familiar with from the gut microbiota analysis, or do different tools apply to measure dysbiosis in the vaginal microbiome?
Yeah, you actually can use various tools. We specifically use some taxonomic criteria, thresholds for specific species. Yep. But it is so almost binary, this state, that almost any diversity measurement tool will work in this specific case.
Wow.
Yeah, so we did these intervention studies. So women that were dysbiotic were screened into a trial. And then we also had a donor program, just as you have done for your company.
Yep.
But in this case, for cervical vaginal secretions that were lactobacillus dominant. And then we dosed women that were dysbiotic with the lactobacillus dominant cervical vaginal secretions. Gotcha. And we tested different protocols, different variations, actually 2 studies, and we just completed a 3rd one as well. And I don't think we have to go into the details here, but the key finding here is that in a significant part of the women, somewhere around 50%, and we've improved it a bit, we can actually get higher now, you will be able to change the microbiome from dysbiosis to lactobacillus dominance, right? And in many cases, that's actually durable. It even stays over cycles. And with this sequencing approach, we also could show that the strains from the donor are actually engrafting. And I think the specific feature of this area is that if you are successful in getting engraftment, you don't get 20% engraftment. No, it gets dominant, right? You switch to this other like ecological equilibrium, this lactobacillus dominance, and you get 80, 90, 95, 99% dominance.
Really?
Yeah. And if you then look at the immune parameters, which was the really exciting thing for us, you actually can see that the inflammation is also turned down. It becomes a immune profile that is very similar to women that are durably already colonized by lactobacilli. So that sort of builds the chain of evidence of causality. And we've used this technology now with dosing almost 100 women. We'll be reporting the 3rd trial. So we did 2. We have done our 3rd trial where we do dose effect analysis and pre-treatments and really to optimize the protocol. And that gives us a very valuable data set. Now, maybe the last thing to mention with this also allowed us, since this is less complex than, for instance, the fecal microbiome, to actually look at the strain level, so not species, but at the strain level, which of these strains do actually engraft in those donors, in those recipients that successfully convert to lactobacillus dominance. And we can do that across different donor-recipient pairs. And then we can go back and see what is the genomic information that actually is driving engraftment. So it gives us something which we refer to as the Engraftome. It's a subset of the totality of genomic information is lactobacilli, but these are the ones that are really potent and engraft. And that information we have used to go back to strains that we isolated from the donors.
Gotcha.
And we've used that to combine to design consortia of specific strains that best mimic, best map, best cover the totality of information of engrafting donor microbiomes. Now that was a lot of words, but
it was. But yeah. I think we're getting it. You're talking about, without putting words in your mouth, sorry if I am, reverse engineering.
Exactly.
The successful transfers to get something which is non-reliant on donors for its future supply.
Yeah.
Which is fascinating. Few questions, if you don't mind, on the concept of vaginal microbiota transplantation. So listeners to this podcast will have heard tons and tons about FMT, IMT, but we've never had a discussion about vaginal microbiota transfer. And it sounds fascinating. Is it like FMT, one donor to one patient, or is it more scalable than that? Do you have to screen the donor for any kind of infectious diseases? how is it administered? So many questions, but just genuinely fascinated.
Yeah, so let me start to try to answer some of your questions, and then we can take it from there. So first of all, the way a program like this, a donor program, is designed is actually very similar. We were very much inspired by the work that's done in the IMT or FMT field. So you screen donors, you check their health, then you look at in our case, at their microbiome, right? Does it have this desired composition? And then you need to do many, many safety tests, right? Because you absolutely want to minimize or prevent the risk of transferring an infectious disease. So that's actually a big part of the activities that, in order to get a program like this running. And then you asked, what does it look like? It is actually a cervical vaginal secretion is collected with a menstrual cup. That is, used more as a collection device, in this case for a CVS material. And we actually did very little processing. So we were able to then freeze this. And then prior to administration, it's thawed and administered with a syringe and a catheter. And it's a relatively simple procedure. And then we've worked on some pre-treatment options of the recipient that actually, seem to facilitate engraftment, and we'll also be reporting out on this. And we've also done some dose response work, which we have not published yet. What I will say is I don't think it has to be one-on-one, but it's also not going to be one donor, 10,000 recipients, right? And of course, one donor can give many, many donations as well.
Gotcha. Oh, totally fascinating. So some people, when they describe FMT, they say the mechanism of action is multifaceted. It's probably driven by bacteria because they're metabolically active, but there's some phage in there. There may be some yeast and fungi. The concept of reverse engineering a successful biological transfer, I think, is, in some respects, it's the kind of holy grail, isn't it, to what this field is it's like kind of like the North Star. But you have other people who, as I say, mention that there's other aspects. So is this the same in the secretions? Are there potentially some other sort of beneficial elements that you're not getting if you have a defined consortia, or are you pretty sure it's bacteria driving the responses that you want to see?
So I think the honest answer that I need to give you is that we don't really know, but we will get the answer relatively soon, right? Because at this moment, our first reverse engineered consortium, we call it FB301, is actually in a phase 1 study where we are addressing a population that was very similar to our vaginal microbiome transplant studies. And we hope to see, of course, a similar effect. I am hopeful for a number of reasons. One is we have looked very, very careful at the data that we got from our transplant studies. And I think we have a reasonably good understanding of what makes a microbe engraft, that there are sequential effects. There's like seem to be sort of pioneering strains followed by others. Yeah. There are some ecological principles appearing on what it makes to be a good colonizing consortium that we, with the best of our knowledge, have tried to re-engineer back into what we have developed. And then the other thing I will say, there are some older studies, other products also that are often single strain, but have shown some efficacy, not enough maybe. But I think this idea that by reducing it to a bacterial component, that you could actually have this effect, there is some pre-existing knowledge here.
And there's reasons to believe.
The reasons to believe, that's the better articulation.
Yeah. So how can you disclose maybe how complex it is? If you can't, that's absolutely fine. But also maybe you could give the listeners your opinion on, within this space, but also maybe the other spaces that you've been involved in, single strain versus consortia. Some people say, well, the single strain is has a number of advantages. It's easily characterizable. The culture, the fermentation, and the drying is more simple. But then others will say single strain's never really going to do the job, unlikely to engraft in enough patients. Consortia overcomes those benefits. So what's your view then on the place of single strains as a former probiotics person, so to speak, and now someone that's developing Consortia?
Yeah, I find it always very difficult to give general answers to this, because I think it will depend on the ecosystem you're targeting, maybe in some cases on the specific biological effect you want to attain. But my sort of overall perspective on this is that I think it will be very hard to, especially if you want to engineer an ecosystem, to achieve that with one strain in almost any human body ecosystem. It just requires multiple activities in most cases. So I think that's probably where it will land. And my sort of intuition says that for the gut, seeing again where the science is now, that, for optimal or close to optimal activities, this will not be one or two strains. This will be larger consortia, which also is and some companies are trying to really, I think, push the limits here on what from a manufacturing perspective is possible, but also what has enough activity. And interestingly, some of these assets are in phase 2 and phase 3 trials now, right? So that will be really, really exciting to see how far this reverse engineered reductionist approach actually works in those indications. Our assumption has been so far that for the vaginal microbiome, you can also get very far with, let's say, under 10 strains in a consortium. But as I often say when I talk about this, you're as credible and as good as your latest clinical data. So we will have to let the data tell us whether we're right.
Fair.
That's where we are now. And so it's a very exciting period for us.
It really sounds like it. So what are the next key milestones then for Freya? Just while we kind of linger on that topic a little bit, what's happening next, and what's your vision for the future?
Yeah, so it's maybe good to say a little bit more on where we are in the development as a company, right? So we started in 2020, like October 2020, we started our activities. And we've put a lot of effort on these VMT studies to show causality, get methods in place, lead us to our first consortium, also do some cross-sectional work to correlate the microbiome in this specific case to pregnancy success rates in IVF. So we have, I think, solid data there that sort of lay the ground for our therapeutic programs.
It's exciting.
And we are now in a phase 1 study with our lead asset, and we're actually, with our Series A raise, which was a substantial one, we raised $50 million USD. We should also be able to finalize a phase 2 trial. Next to that, I think it's also good to mention that, which we announced fall last year, we've received an investment from the Bill and Melinda Gates Foundation to work on a second asset that very specifically targets bacterial vaginosis and some of the bacterial vaginosis features that you also see in low and middle income countries. So that's a second asset we have under development, and that should also go into a phase 1 under the current financing period. And then, of course, we are always thinking about possibilities to expand our pipeline, maybe with additional funding or partnerships. So there's a lot of activity ongoing at the moment. It's an exciting period. It
sounds like it's one of those really, really exciting chapters where you've got data coming, you've got enough cash to get through the next inflections. And I guess it's just about delivery and really the modality doing the magic and hopefully generating some good clinical data.
Exactly.
So we wish you all the best.
About excellence in execution also, right? The pressure is on us. That's also how we feel as a team.
But that's probably healthy, right? So we've talked about Freya. When you look at your resume, you've actually been involved in a lot of entrepreneurial type activity. and you've been involved in other companies as well in the microbiome space. One of the things that would be really, I think, quite cool to discuss is the Kaleido journey. And for the listener, Kaleido was one of the companies that appeared when the microbiome was at its hottest. Can I say that?
Yeah.
You'll tell me how much money was raised, but it was a lot. I just wondered if you could tell the listeners a little bit more about Kaleido and also some of the lessons that might be quite interesting to hear, particularly for those that are involved in companies at the moment.
Yeah. I'm absolutely happy to share some things about this. So let's maybe use a few words about what Kaleido did. Kaleido developed what we call synthetic glycans, essentially sugar polymers, but in a very targeted fashion, right? So it's chemically synthesized, not extracted, which allows you to build specific molecules. And then these molecules were analyzed for their ability to feed specific microbes in the gut microbiome in order to amplify their presence or their metabolic activity and their impact on the overall ecosystem and the host. That company, I was for a while chair of the scientific advisory board, and after they did an IPO, so went public, I actually joined as chief scientific officer. And as you allude to that, I think that the story has two sides, right? First, I want to say that the technology actually, I think is still very exciting, and is a proven technology, and it also led to some, really scientific papers that I'm personally actually very proud of. I think still are impactful for the field. So there's a paper in Nature Communications that describes how it was synthesized, how we built it up, the screening platform, and that I think is valuable for the field. And then actually last year, a little bit late, but we published also a paper with Jeff Gordon, who then analyzed it in mice models. And this paper describes, it's in Nature Chemical Biology, I think, that really describes the how it favors the specific Bacteroides species in this specific case, and gives very detailed mechanistic understanding, also with some cool technological analytical tricks. But that's for people to read. So,
is it? Forgive me if this is a really, as they say in Scotland, daft laddie question. Is it a prebiotic?
It is. It has the prebiotic concept, but it's a level of refinement that you do not get if you extract carbohydrates, right? Because if you extract it from plant material, you're basically a bit limited to what the genetics of the plant allow it to synthesize. And then there's also lots of variability, like where do you grow it? What was the season?
Yeah.
So it's less variation. It's less ability to target specific structures and therefore taxa. So it is, it's taking it to another level of precision. And then this advantage of, in principle, endless scalability, I think is also very attractive if you do it in a synthetic way. So it has natural sugars, but like synthetic polymers. That's the idea.
Gotcha.
So what happened to Kaleido? I joined, as I said, after the IPO as a chief scientific officer. And at that time, the company was also running, drug trials, phase 2 trials. I think one of the, unfortunate things is that maybe the indications that were chosen to develop as a first lead product were not the most, not the best ones, in the sense of difficulty to show a clinical benefit, and also from a market patient perspective, also not optimal. And I think that was particularly difficult at a moment that the company needed to raise more money in a time where there was a COVID pandemic. So it was really hard to do clinical trials. And also at that time, actually, the financing market for the biotech industry really was very bad. So, that in the end led to the fall of the company. which is unfortunate. It's not fun to be part of, I can tell you.
Yeah.
There's many things that you see when something like this happens that we can talk about it maybe once over a beer at a conference, but we're not going to do that now.
Yeah.
But I do think there are a few important learnings from this, and that's probably what you're trying to look for. For me, it means do your work really, really well at the start, right? It's not only about the cool technology, but make sure that the science and the clinical rationale for what you want to develop is really, really well thought through.
Makes sense.
Right?
Yep.
Then the second thing is the clinical development path, right? What clinical trials do you need to do? What's the regulatory context? What data will you generate if you do your next raise? If you raise 30 million, can you generate clinical data that are valuable enough for people to put more money into this? That all has to be lined up. Then, of course, you need to have a real business case on the unmet medical need and who's going to pay for the product in the end. And then the fourth thing is think very carefully about manufacturing. What will it look like? What will it cost? Is it scalable? Who can actually do it in the end? Those are sort of the four legs of the stool. And then on that stool, I think you need to have a excellent team that is not too bloated in the sense don't grow too big, but that has these core expertises that can really execute on the plan. And there's just some things that I take from this period, like where maybe not all of these legs of the stool were carefully enough analyzed, and also this fact, if you have a lot of money, to grow very quickly a big team is also something that is tempting because you can do so many things. But you need to do not what you can do, but what you absolutely need to do. And maybe not more at a certain moment.
And also being in the US, probably much more expensive, right? If you've got a big C-suite.
Yeah. That's true. But I will also tell you one thing. I think where we in Europe are maybe less tempted by growing too fast and being too ambitious, there's also a flip side of this. I really have always enjoyed the Boston biotech ecosystem because there is so much intellectual power there on such a small amount of square miles. There's so much willingness to take risk and to actually try things. There is the money system is also set up to actually allocate risk capital and just try to some extent. So that's also very inspiring. It's not only, it has certainly not been a negative experience for me, and I still have many, many close and very good contacts with people from in the company and around the company today.
Yeah, certainly sounds like, although it wasn't the outcome that you were all hoping for, it's been a great journey for you to go on that you're now applying to all the things that you're doing at the moment, which, on balance, the positive thing.
I will maybe one thing.
Please.
The technology is not lost, so it's still developed more by a company in the food supplement area. And actually, I'm advisor of a company that licensed back one of the lead assets.
Yeah.
You can find that information also on the web. So it's called Sidekick Bio. I'm not driving any of this, but we're trying to give it a second chance. And I think there's a good plan also for making this happen.
Because the science is good, so
the science is
you should give it a go. I'll just add a couple of comments on the Boston side. I feel the same way every time I go over there. The energy, the ambition. To be frank, the American dream is a thing. It's real. And I feel it every time I go over there. People want to be the best. They want to be first. They want to change the world. Everyone wants to make money. They want to help patients at the same time. And I think we do like that a little bit in Europe. And we're competing for capital with those companies. So maybe there's something there.
I think it's very well said. And, one of the things, this is more like a personal story. I've worked in France, in Denmark, in the Netherlands, in the US. Every time I move to a new country, you learn a lot. And it's also humbling, actually, because a lot of the assumptions you had on how things should be or what good actually looks like, they are sometimes challenged by this, right? And this aspect of the relentless ambition, the also not being embarrassed to express your ambition and just go for it, combined with the the crazy amount of talent that you have there is also a really driving thing. It's not a coincidence that many of the big tech and biotech successes come out of the US. There's, of course, many things that fail.
Yeah.
But if you have a good idea, it can also grow very fast there.
Yeah, 100%. Every time I go over there, I feel energized. I think, wow, because to be honest with you, in Scotland, sorry, but there's probably a handful of clinical stage drug development companies. Really, you can probably count them on one hand, whereas you walk on one floor in one of these buildings in Boston and there's probably about 40. It's crazy. So
the anecdote I often tell is,
please.
My wife and I, on a Friday afternoon, we often went to one of the local breweries to just, have some time together and evaluate the week. And without exception, you were surrounded around you with people that were talking about their next idea.
Yeah.
In tech, in biotech.
I love it.
And that is really true. And it's inspiring. Yeah.
Yeah, I love it. I think it's nice you've had some time over there. My wife and I would love to go as well at some point. Mm-hmm. We hope to. And then maybe we'll end up in the sun one day like you. But for now, we're trying to build a global company here in Scotland. So there's just a couple of things I wanted. We've been going for roughly an hour now, by the way. Time flies. It's been super interesting. There's a couple of things I did just want to touch on before we proceed to wrap up. One of them was the Gates Foundation, which you mentioned. And you're involved with the foundation, I think, in a kind of advisory capacity. And also, you've raised some money from them. I don't think people listening know that much about the foundation, and I think it could be quite interesting for them to hear about the scale and the power and their ambitions in the microbiome and the developing world, because it's unique, I think, globally, isn't it? And also how it's been for you being involved in this. That could be pretty interesting.
Yeah. So I want to be very mindful. I'm not a representative of the Gates Foundation, so I can just share some of my experiences and why I've actually been find it inspiring to help them achieve their mission. So I will say I think many of us have worked at some stage with public grants or NGO funded grants. And they often have very large ambitions to solve some of the major problems, big clinical problems, poverty. But I think what is often difficult is that many of these programs, you maybe get a grant for 3 years and then you have to hope you can continue. Yeah. And I think Europe has some sort of system to try to solve this, but it's maybe not always very fast and predictable, right? What I really like about the work of the Gates Foundations, and they literally invest billions and billions per year in solving some of the big challenges in low and middle income countries. And the way they do this is they start with fundamental science in many cases. They fund the fundamental science, but then they take it all the way to clinical interventions, product launches, right? And I think there's hardly any other organization that does this. And this whole culture of quality, but also translating, making products, a program is not successful if it doesn't lead to that, I think is pretty unique. So my role there is really, really modest. I want to be sure that people realize this. I have an advisory role, right, in one of their programs, or one of their many, many programs. But I always find it very inspiring, and I hope I can, give some help with the expertise I have specifically in my area of in my background. And then the other thing you mentioned is that Freya has not received a grant, but actually an equity investment. They also very rarely do this in companies, which I think we take as a sign of their appreciation. It's an endorsement. It's not only money; it's also an endorsement.
It's an endorsement.
And again, it also puts the pressure on us to deliver. But we're very fortunate to have this. Yeah.
So why don't we kind of proceed to the wrap up, and maybe you can give the listener some. Can I ask you for some predictions about where you think the field is going to go? What's going to happen in the next 3 years in microbiome field more generally, and maybe give us some reasons to be excited.
Yeah. So yes, we should be excited, right? Because the core, we've talked about this, the core is there. This has so much potential. The science and the clinical relevance, I think, is sort of validated. Then the other thing I will say is we also live in a very uncertain world, right? For many aspects. One of them actually also really touches upon the drug development domain. And without deliberating too much on that, I think we all need to see and understand what will happen in the US environment and how that impacts the rest of the world. I do want to actually maybe allude to one point that we haven't touched upon. I'm also part of what is called the Microbiome Therapeutics Innovation Group in the leadership there. And we try to be a very active player. And essentially, the activities focus with the other companies that are, many of the other companies that are developing microbiome therapeutics. We try to align with regulatory bodies, specifically the FDA, on the regulatory path and making it as smooth and fast, but also high quality as possible. There's a lot of outreach to policymakers, and then it's education to KOLs, but also all kinds of other organizations. I think it's important to mention that because we as a field need also to have a collective voice. And so I'm very dedicated to make that happen. So where do I think this will be in 3 years? There are a number of clinical programs at the important companies, one of them being us, that should have had readouts, right? Clinical readouts in phase 2 or phase 3 trials. So I would be very, very disappointed if not some of them would be positive. And I think if just one or two give a really good result in indications with a big market potential, that could actually rapidly change the sentiment around the field. And I will also say maybe one thing more. If you see how, especially in the US, but also in other areas, how people start to articulate maybe what is important for the future therapeutic field, there's some concern about us trying to address diseases with very expensive medication, proprietary small groups that actually maybe also leads to long-term adherence and is very costly. While on the other hand, you can see that with microbiome therapies, often you can actually address the root cause, right? We talked about inflammation. If you can take the signaling out, so you don't need the other drugs to treat the symptoms.
Yep.
So I think in that respect, it seems that our therapeutic area is also positioned well in where maybe drug development and treatment paradigms as a whole are going to move. So we should feel this as a tailwind, I would say. And I hope that we continue to get good results and execute with excellence. We should be set up for an interesting and a positive outlook.
Good. Well, thank you so much for your wisdom, for your insights, and then for the encouragement and the positivity there towards the end. That was awesome. We've covered so much. We've talked about women's health. We've talked about fundraising, company creation, company building, clinical development. It was exactly what I was hoping for. So on behalf of all the listeners and me as the host, wanted to thank you for your time and wish you all the very best with all of your endeavors.
Thank you so much. It's been a real pleasure. So hope to see you in person soon.
I hope so. Thank you.